Okay, so I propose we start. Dear audience, thank you for joining this webinar. We are very happy to hear Avi Sadka talking about polar auxin transport and alternate bearing in fruit trees. So we are in a Journal of Experimental Botany series organized by the Society of Experimental Botany. And before giving the lead to Avi, I wanted to give you some information about his career. So as you may know, Avi is a plant physiologist. He works at the Volcani Center in Israel in the Department of Fruit Trees. He did his PhD at the Weizmann Institute in Tel Aviv. And then he went to Texas, USA for a postdoc and came back as a researcher at the Volcani Center. And Avi is also teaching at the Hebrew University. And since he did 2 sabbaticals in a group of Eduardo Blumwald in Davis University in the US. So Avi, we are very pleased to hear about this very interesting work that was recently published in JXB.
Thank you very much, Yves, for the nice introduction. And I also thank the organizers for the invitation and for the opportunity to present our work. So alternate bearing, the definition is that heavy fruit load 1 year inhibits next year flowering, which is defined as return bloom or back flowering. And it is quite common among many fruit trees or in many cultivars of most fruit trees. This very old, this scheme taken from an old review paper. And what we can see that climatic triggers such as spring frost or spring heat wave synchronized yield at the orchard region or even country level. So climatic trigger can generate also alternate bearing in regular bearing cultivars. But as you can see, if it is not repeated, the climatic trigger, it is mitigated throughout the year. And there's just to show you that sometimes we can find, this is a picture, Mandarin photos of 2 trees, the same orchard, taken the same day. And you can see that one tree bears a lot of flowers, while the other one is empty, almost empty of any flowers. And this is the same for olive. We can see a branch during the off-crop year, and a branch during the on-crop year. These are real data taken from avocado cultivation in Israel, which started in the mid 70s, and we can see the green bars are the area. So you can see that in the last years there is an increase due to a demand in avocado in the world market. And red line represent the yield in ton per dunam. Dunam is 1/10 of a hectare, and we can define here, in fact, two periods. In the first period, where cultivation practices were relatively, I would say, primitive or not that developed, where the yield was less than 1 ton per dunam, we can see non-regular alternation in yield. But as soon as practices developed and yield exceeded 1.6 to 1.8 ton per dunam, we can see the fluctuation, the yearly fluctuation in yield, which is caused by the relatively heavy fruit load. So an on year would be followed by an off year, and so on. These are real numbers, so you can see that alternate bearing has a economical impact on grower income. I would say that during the on year, the fruit tend to be small, and sometimes not as profitable as required. So we are talking, in fact, this, as I mentioned, this is a natural phenomena. And in fact, regular beering and alternate beering are two strategies which are interesting. And we can view alternate beering in an evolutionary view. And there are some indication that maybe domestication forced for regular beering. We have natural occurring or wild fruit trees, which in the wild are more alternate bearers. But when they are forced into domestication through cultivation or through breeding, they become regular bearers. And the other thing, the other interesting point is that maybe alternate bearing in tree perspective is more economically is more reasonable. Why do I say it? Because in most subtropical fruit trees, flowering occurs on 1-year-old shoots. So vegetation is required for flowering. However, overvegetation competes with productivity. So regular bearer tree would require to allocate its resources proportionally between vegetation and flowering, while the alternate bearing avoid this dilemma by allocating its resources either to vegetation or productivity. So it's a reasonable strategy. But our research does not deal with this kind of questions. I just wanted to mention it. So if to summarize this part, the heavy fruit load both does two things. It reduces vegetation and it reduces flowering. But we just saw that vegetation is required to flowering. So the reduced flowering occurs also due to reduced vegetation. What are the stages that might be affected by the heavy fruit load? So classically, we are talking about flowering induction. So flowering induction is inhibited by the heavy fruit load. However, in citrus, as in other cultivars, other stages of flower development also might be affected by the heavy fruit load, such as evocation, differentiation, and bud break. And this is dependent on fruit presence on the tree during these stages. And here is the scheme which explain it. So this is the citrus annual cycle in the Northern Hemisphere, of course. So during the November, this is the bud, a small organ in the base of the leaf. And when it receives the flowering induction signal, it can, of course, it will give rise to inflorescence. However, if it does not receive the signal, it will develop into vegetative shoot, okay, which everything occurs, of course, during the spring. And then following flowering and fruit set, there's fruit development, stage 1 and stage 2, and then maturation of the fruit. So maturation of early season varieties, such as Satsuma mandarin, occurs already during September and October. And then most citrus variety, their maturation would occur during the flowering induction period. And we have many other varieties, which are very late varieties, such as Murcot mandarin, Valencia orange, and many others, which their maturation occurs even when flowers and even fruitlets are on the trees. And the general scheme is that these cultivars would tend to be more alternate bearers than these cultivars. However, this is a very general rule, and there are many, many exceptions for this rule. The major external inductive condition for flowering is low temperature or accumulation of cold hours during the fall and the winter. And now you understand why global warming and warmer winters are so harmful for productivity in subtropical fruits. And it might be assumed that the heavy fruit load generates an inhibitory signal, which we define as AB, or alternate bearing signal, which blocks flowering induction. And in fact, why do we think it? Because when we remove flower or fruit until the mid-October, we can induce back flowering during the next autumn. And this is a result taken from a Spanish group. We can see that when fruit was removed during August, September, and October, before the onset of the flowering induction period, back flowering or next year flowering was as normal. However, when fruit was removed during November and on, back flowering was not achieved. So we don't know, or the aim of the work was to find the nature of the alternate bearing signal, but we concentrated in our work on what is going on in the bud, because the bud is the organ that needs to take the decision whether to go to vegetation or to reproduction. And this is the question that we initially asked: what is altered in the bud? And obviously the approach, or before the approach that we took, which was a genomic approach, this is an interesting physiological experiment which was not published, and it talks about the memory in the bud collected from on-crop trees. So what we did in this experiment, we took small stems just before the onset of the flowering induction period, and we put them on synthetic media, which contains, of course, sucrose, under temperature permissive conditions, which are low temperature. Now, within 2 weeks or 3 weeks, the buds which are on those short stems would break. Some of them would break into vegetative shoots, and some of them would break into flowers. And these are the numbers. So when the shoots are taken from an off-crop tree, we see that most of the bud would develop into vegetative shoot. This is not normal. It's not like what happens in the tree, but still about 20% would develop into flowers. And you can see that about 15% would not develop at all and would die. Look what happened when we took those stems from on-crop trees. In fact, more than 80% of them simply die, and we get no flowers. So this experiment demonstrates that the bud bring with it a strong memory of the fruit load. And it simply, even under such a permissive conditions, when resources are not limited, it simply die because of the memory. Now, what we did a few transcriptomic analysis, and here I present only one, which was performed by Liron and Sivan, which were graduate students in the lab. Don't underestimate the importance of this guy in the work. And what we did here, we removed, or what Liron and Sivan did, they removed the fruit around mid-August, and they collected the buds after 1 week, 2 weeks, and 4 weeks. Okay, they did a full fruit removal, the fruiting, and we have, of course, the off-crop after at time 0, after 4 weeks, we have the off-crop buds as controlled. And look what happens. Within 1 week after fruit removal, the on-crop buds, which their fruits were completely removed, became, in terms of their gene expression or transcriptome, they became like off-crop buds. And this was a very dramatic result. Now there were many processes that were changed. Here I'll show you some data. So this is the expression of flowering control genes in on and off crop buds. And for instance, if we look at Apetala 1, FT2, which is the canonical FT, leafy, we can see that toward flowering induction period, these three genes are induced. And in our transcriptomic data, we observed an increase in their expression within 1 week after fruit removal. So we can see that FT2 is induced, SPL5 is induced, and so is leafy. Now, one of the most striking processes that was altered in the buds following fruit removal was a process which was calcium dependent and independent auxin homeostasis. This is a scheme taken from a recent review by Leah Band, and here she summarizes the polar auxin transports across membrane, which is mediated through a few membrane proteins such as spins, OX1 lax, and ABCB membrane proteins. And as you can see, and this is a relatively recent finding, auxin transport can also mediated between one cell to the other by diffusion through plasmodesmata. And we know that we can block the with compounds like NPA, TIBA, and PBA, we can block the X-auxin efflux and auxin movement between cells. Here are some of the transcriptomic data. So what are the genes which genes we show were altered following fruit removal? So I just mark here, these are all calcium dependent genes, and I mark 4 genes which belong to one family, which is called PBP1-like. And 4 of those genes were remarkably induced in the off-crop buds relative to on-crop buds and following the fruiting. What is the PPB1 gene? So this gene was demonstrated in Arabidopsis many years ago that it has to do with it's called pinoid binding protein, as the name suggests, it has to do with pin protein regulation, and it is, as I'll show in a minute, has to do with calcium-dependent polar auxin transport. Another gene, which was called NPH1 for non-phototropic hypocotyl gene, was also remarkably induced following fruit removal. And again, previous works in Arabidopsis show that this gene is also involved in auxin homeostasis or polar auxin transport. And in this scheme, I summarize the role of these 2 genes. So we know that auxin homeostasis takes care through the pin proteins and other membrane proteins, either inside the cell or outside the cell. Calcium homeostasis is mediated either through carriers, antiporter carriers, which are called CACs, or through calcium channel. Now, what it was found in the literature that PBP1 bind calcium following, and then it become activated. It activates a gene or a protein, which is called PID, which is responsible for the activation of pins. So this is the connection between calcium homeostasis and auxin polar transport. But what I mentioned, that there is another gene, or another protein, NPH3, which is can also activate pin, which become active in auxin polar transport. And how these genes looks in our transcriptomic data. So this is the CACs. You can see a remarkable induction in CACs protein following fruit removal, and 2 members of the PBP genes, which are also remarkably induced, and this is an NPH3. so remarkable induction. And also another calcium binding protein, which has nothing to do with auxin transport, auxin polar transport. So the question that came from this work is all auxin polar transport activated in the bud following fruit removal. So what we did next was to examine auxin levels in the bud following fruit removal. And we can see very clearly that following fruit removal after 1 week and after 4 weeks, there is a remarkable reduction in auxin level in the bud. And next, this is an immune localization using anti-IAA antibodies performed by Yasmin, another graduate student. And we can see very clearly these are 2 on-crop meristem or on-crop buds. And we can see the labeling of the auxin. I'm sorry, these are the 2 on buds. And we can see the auxin labeling in the apical meristem, which is hardly detected or non-detected in the off-crop buds. So this is in line with our metabolomic analysis of auxin levels in the bud. All this reminded us of a very relatively old hypothesis, which was raised by a great German physiologist, Fritz Wangert, at the end of the 80s. And Fritz talked about primogenic dominance. We are used to talk or are used to think on apical dominance. But what Fritz defined is that there are other types of dominance, such as primogenic dominance. And here, what the example is the first developed sink, which, of course, a lot of the photo assimilated are from the source are translocated into this sink. So it became dominant over the other sinks. Okay. Now, I want to give you an example. So in citrus, for instance, we can see that the fruit is in apical position to the buds. So this is a classical apical dominance. However, in olive, it's a different story, because in olive, the first year branch or the part of the branch which grows during year 1, which has fruits, is dominant or exerts a dominance of over the second year growth, which is fruitless. So this is clearly not an apical dominance, but a basal dominance. So if we come back to Wangert's suggestion, what Wangert suggested that auxin flow out of SIC1, because it's dominant, is stronger than the auxin flow from the other sink, S2, S3, and S4, whatever. And this strong auxin flow inhibits auxin flow out of these sinks, okay? And this is, in fact, the mechanism for the dominance. And the question that we ask, does the block auxin flow from on-crop buds play a role in uttered bearing, signaling, or control? And now I come to the paper which was published recently in Journal of Experimental Botany, where we try to answer the question whether auxin polar transport indeed plays a role in the signaling of the heavy fruit load. And the work was initiated by Liron, again, a graduate student in the lab. And for that, Liron used radioactive auxin, which was applied to a donor agar on top of either an on-crop or the fruited or off-crop stem, okay? And what Liron did, he measured how much auxin, or what is the auxin polar transport between the donor agar to the receiver agar through the stem. And this is defined as polar auxin transport. Now, the result was very clear. We can see that following fruit removal, there is a significant reduction in polar auxin transport in the stem, again, following fruit removal. This is the polar auxin transport in the control, in the presence of the fruit, and this is after fruit removal. And this is now with olive, the first question that we want to ask is polar transport going to be apical to basal or basal to apical? And what we did, we used either the donor or auxin was applied apically or basally to the new shoot. And this was performed by another graduate student in the lab, Dor Chaim, and the result was very clear. This is auxin polar transport when the auxin is applied at the basal of the shoot. And you can see that when the auxin is applied to the apical donor, there's hardly any polar auxin transport. So the answer is very clear. Bud is generated from fruit direction regardless to its position to the bud, apical or basal. And next, like in citrus, we can see that fruit presence generate strong bud in the shoot, and in the off-crop shoot, of course, that the bud is greatly reduced. So, if to summarize until now, the fruit generates a strong IAA polar transport in the stem. And now the question is, what happens? I remind you that Banger suggested that this strong auxin polar transport in the stem would inhibit auxin release from the bud. So what we did next, again, using radioactive auxin, we applied the auxin, or Dor applied the auxin into the bud directly, okay? Either to an off-crop stem or to a defruited or off-crop stem, okay? And the results were very clear. When the fruited stems were used, we can see a great reduction in the radioactive auxin in the bud and its release to the stem. So fruit removal, which reduces auxin polar transport in the stem, would induce release of auxin from the bud, exactly according to the hypothesis. The same is with olive. So again, like in citrus, fruit removal in olive stem induces auxin release from the bud into the stem. So the next question that we asked, can auxin replace the fruit? So what we did here, we applied to an off-crop stem, we applied for 1 month called IAA. Okay, just to mimic, because the idea is that the fruit generates a strong auxin polar transport in the stem. So we took an off-crop stem and we applied to the donor agar a cold IAA. And then we applied during the experiment the hot or the radioactive IAA to the bud itself. And the results was very clear. So clearly when auxin was applied, a cold auxin was applied continuously to the stem, it blocked auxin release from the bud, okay? So it's exactly the same as the control, which is the on-crop stem, okay? And this is again, when the off-crop bud, we can see the release of auxin into the stem. So if to summarize with this scheme, so this show stem with a fruit, or with heavy fruit load. And we can see that the fruit generates a strong, or a strong polar auxin transport, which blocks, in fact, the auxin release from the bud, okay? And the result is induction in auxin level in the bud. Now, the next question, which I don't have yet an answer, how this is connected to flowering inhibition. Is auxin induction in the bud has some negative effect on flowering? This is an open question. Truly, we couldn't find very strong indication in the literature that auxin plays a role in flowering induction or flowering inhibition. And this is a very interesting question, whether it does it directly or indirectly. And now we're trying to answer this question. Just to show you a very simple-minded experiment, field experiment, we applied auxin to off-crop trees, and the result was a 50% reduction in flowering. It's not very dramatic, only 15%, and we think that this is due to a very efficient mechanism of auxin removal from the plant. We know that auxin cannot stay in excess in plant organ or plant tissue, and there is a very efficient way to remove it. And so the effect that we get in the field is only 50% reduction in auxin level. Now, just to summarize this report, so is the auxin polar auxin transport is unique for fruit tree or for signaling the heavy fruit load to the bud? The question is no, and this is a work that was performed in the last few years in Tom Bennett's lab, and Tom talks about the phenomenon in Arabidopsis, which is called the end of flowering, and how it is controlled. And so you know that this is the life cycle of the Arabidopsis plant. There's vegetative development, which is followed by reproductive development, and then cessation of flowering, which is defined as end of flowering. And this is a highly controlled, it's not just that flowering stop, it's a highly, what Tom shows, that this is a highly controlled phenomenon. And again, auxin polar transport plays an important role in end of flowering, because what happens is that the developed calyx generate a strong auxin flow in the stem, which inhibits canalization of the auxin from the new flowers or from the buds. And this generates an inhibitory signal, which in fact caused the end of flowering. So I will end my talk by thanking the people who in my lab that were involved in the work. I've mentioned Liron, Dor, and Sivan, and Yasmin, which were graduate student, and Mila is my technician. And the work was funded by the Chief Scientist Fund from the Ministry of Agriculture and Rural Development. And thank you very much for listening to my talk.
Thank you very much, Avi, for this very clear presentation. Really a great talk. Do we have questions from the audience? You can also write down your questions in the chat. Okay, if there's no question, maybe you can find other questions. So I will start with the questions. So you mentioned at the beginning of your talk that the bearing of fruits, the alternate bearing, one physiological reason could be that the trees would exhaust their reserves, especially carbon reserves. And so this suggests that there might be a link between carbon metabolism and auxin signaling. And as you know, I guess there has been recently findings about the link via sucrose and ferrous 6-phosphate signaling. There's also strigolactones involved in cytokinase. So how do you put that into your thematic?
Okay, so one of the things we in our transcriptomic data we show we saw some differences in trehalose phosphate metabolism. We did some experiments, some physiological experiments, I mean, in the right direction. We did some experiments. We tried to manipulate the level of trehalose 6-phosphate, but we failed. I mean, we couldn't get any reasonable, or the data were not consistent. So I don't know, but clearly one of the directions that we are now checking is strigolactones, which are, of course, are involved, were recently shown to be involved in dominance or apical dominance. So this is one of the directions.
Okay, so I see there's a question from Mareike Jezek. If you want to ask a question, or I can read it as you want.
Yeah, so exogenous application of gibberellic acid can also prevent flowering. Could there be an interaction between auxin and gibberellin signaling in the buds? So obviously this was our hypothesis. We thought that, so one of the things, the interesting things is related to the memory of the fruit. I mentioned that we can block the memory if we remove fruits about 1 month prior to flowering induction period. Now with olive, we have to do this much earlier. So fruit memory is much longer in olive. So if you remove the fruit, say in August, this is the last, in fact, the last time, or the beginning of August, that's the last time to get back flowering. And we thought this is to compare between olive and citrus. This is a great, way to answer the question of the memory. And in fact, our hypothesis was that what generates the memory is GA in the buds. So we thought that when the memory is reversed when GA is not induced in the bud. This was our hypothesis. So as long as GA, so what we thought, because there are indications in the literature that auxin, for instance, in fruitlets, or during a fruit set, it is well known that auxin can induce gibberellin biosynthesis. So our idea was that auxin is high in the bud because of fruit presence, but as long as gibberellin is not high, we can reverse the memory. And as long when GA is induced, it's you cannot do it anymore. The data is not very strong in favor of this hypothesis. I still think it is valid that auxin can induce gibberellin biosynthesis in the bud or in the plant. However, I can just say that it's not, the results are not conclusive. For 1 year, we saw indeed an induction in bud gibberellin, but in another year, when we took the buds from the orchards, we couldn't see any effect. So it's a good hypothesis, it's a good idea, but probably further work needs to be done in order to prove it. And one of the problem is that there are only very few labs, maybe 2 labs in the entire world that can perform a good gibberellin quantification. You know, the gibberellin there are many of them. It's very complicated, and the expertise of doing gibberellic acid metabolic profiling is quite difficult.
Yeah. So if there's no questions, I have another question.
Avi, may I ask a question?
Sorry. Yes, go on. Yeah.
May I ask a question? I would like to know regarding the treatment that, sorry about not being able to see me because I tried to start the video, but it says that your video cannot be started because the host stopped it. So this is why I'm shut down. So regarding the application of auxin, have you tried to do successive application of auxin to try to see whether it's okay? And to try to see whether you can reduce this
the flowering.
Yeah.
So I think that what we did in this experiment was 3 applications of auxin, if I'm not mistaken. And
so what you're showing is after 3 application of auxin?
I think so. If I'm not mistaken, I think. But again, don't forget that the plant is a very, very efficient way, either by conjugation or catabolism, to remove excess of auxin. It's a very fast response. So it's very difficult to induce auxin levels. And you know, we try even to do so. We applied. In the greenhouse, we applied auxin directly to the bud, and we could never get a more significant effect.
It would be interesting to test whether you have an increase in auxin conjugate following the application, because that might be one of the effect that you.
I didn't show it, but in the bud following fruit, I just saw the free auxin, but in the bud following fruit removal, we see also induction in auxin conjugates. I just didn't show the result.
Okay.
But it's not after auxin application. Yeah.
Okay.
Do we have another question? Okay, so in your article, you mentioned, if I remember correctly, that auxins could repress cold induction in the bud. So what is known about cold induction? How does it work? And how would auxin interfere in there?
Truly, well, we know that there is in Arabidopsis there is of course the vernalization dependent pathway, which is very well characterized. In fruit trees, I just wrote a big review about alternate bearing in fruit trees, and I was trying to fish some solid data from fruit trees. There are some indications that homologous genes are induced during cold treatment, so there are only indications, but in the connection to auxin, I cannot answer it. We don't know.
Okay, so because I think it's still mysterious whether epigenetics or proteins accumulating or metabolic signals, ABA might be involved.
So regarding I should mention it that regarding fruit memory, there is a work by a Spanish group that shows that epigenetics or FT epigenetic modification of FT promoter plays a role in generating the signal of the heavy fruit load. So probably there are few mechanisms or few pathways which leads to flowering repression by the heavy fruit load. But clearly, epigenetics play a role. It's a nice work that was published about a year ago in a New Phytologist.
Yeah, thank you. Do we have another question? We have still a few minutes. So let's ask another question. How do you see the translation of your work into the world of fruit production?
Oh, it's difficult, of course. It's a basically a basic work. There are, in avocado, for instance, there is a report by an American citrus physiologist. She claims that she applied, I'm not mistaken, very right, she claimed that she applied auxin polar transport inhibitors, TIBA, and she could affect flowering. I mean, if you apply it, of course, to the stem and you block strong auxin polar from the stem, then you allow release from the buds, and this should affect flowering. But I don't recall seeing any scientific paper. This was just, some briefing from the news briefing or something like this. Verd, am I mistaken?
The application of TIBA was together with cytokinin, so it might be that what it does, it's convert the cytokinin to auxin ratio, increasing the cytokinin. So this is one of the theories, at least in avocado.
Thank you.
Okay, so I think time is running out. So I think we can applaud again Avi for the very nice presentation and the very interesting discussion. And there is also a message in the chat. As you may have seen, you can you have a survey to give you feedback about the seminar, the webinar.
Thank you.
Okay, thank you very much, and bye bye.
Bye bye.